So, okay, I think we will begin. Can everybody see the slides okay? Yeah. So, good morning, everyone. Welcome to this Alliance Exchange webinar on Thinking Differently, an introduction to exploring non-animal approaches. My name's Juliet Dukes and I'm Research Manager at Frame, and I'm presenting this on behalf of our organisation, but also Dr. Michelle Hudson-Shaw has been integral to the development of this area of work. This session is being recorded, as I mentioned earlier, and we will make it available on our YouTube channel in the near future as well. So we get started. Just briefly, at the Fund for the Replacement of Animals in Medical Experiments, or FRAME, we do exactly what it says in our name. We fund research through our summer studentship programme, paying for undergrads to carry out their own research projects. We fund small grants programme for pilot and proof of principle studies under the Innovation Grant Scheme. And we also fund the FRAME Lab at the University of Nottingham's Medical School. We share information through our own journal, Atla, via our website, social media, articles and events. And we also create change through influencing policy by engaging mostly with regulators, so the Animals and Science Regulation Unit of the Home Office, and we also brief MPs and provide expert response to consultations and queries. We have a strong background in running training schools at Frame, and mostly in experimental design and statistics, and we run these nationally and internationally, focused primarily at postgraduates and early career researchers. So today, I'm going to present some of the work we're doing aimed at encouraging researchers and other stakeholders within the animal research community to think differently about their research and help them transition away from animal use. We'll start with some background and a current look at the landscape, how we found out what's needed and what would be helpful to the community, and then we'll have a look at some of the content that we're producing and think about the next steps. I'll hopefully only be presenting for about half an hour, so we should have 15 or so minutes for questions afterwards. So please pop any questions you have into the Q&A channel or the comments. So recent events, the past 12 months has seen quite a lot of noise around the use of animals in science in the UK, and that noise hasn't come from the usual suspects. So in June of this year, Professor David Main, chair of the Animals and Science Committee, the independent advisory group to the Home Office's Animals and Science Regulation Unit, penned this letter about the strategic direction of animals and science. And he states that he believes that the UK is at a significant juncture in the required use of animals in science and the drive to accelerate the development and uptake of new non-animal technologies, NATs, and new approach methodologies, NAMs, including those relevant to regulatory testing. There are significant potential scientific and economic opportunities associated with these innovations. Other countries are now making plans and progress for promoting non-animal alternatives, and David Main believes that it's time for the UK to take the initiative. So that was really progressive. We also had formal acknowledgement that our current framework and processes are not doing the job they're supposed to. In October last year, a review of the current antibody licenses, so 31 of those in total, all granted over the past 5 years, showed that many licenses did not provide sufficient demonstration that they had actively searched for third-party suppliers and libraries to confirm whether the antibody they need is already available. They hadn't provided sufficient demonstration that the antibodies cannot be isolated from third-party animal-free libraries, nor have they provided sufficient demonstration that it's not currently scientifically possible for the antibodies to be produced by non-animal methods. So clearly something is missing here. Sorry. We also saw this year that review processes are failing to address replacement. So in February, there was a publication of a report carried out by Dr. Francis Rawls as commissioned by the NC3Rs. Dr. Rawls interviewed and surveyed stakeholders within the animal research community, from animal license holders to Home Office inspectors and funders, and found that replacement does not seem to be covered well by any of the review processes. Establishments should ensure that their processes allow the use of animals to be challenged earlier on in the research planning process, and this could be facilitated by guidance to AWERBs on questions to ask and what should reasonably be expected of applicants. Also, she noted that funders' peer review process involves more specialist scientific expertise, but their review tends not to focus explicitly on whether suitable replacements might be available. We also had the first guidance from the Animals and Science Regulation Unit for project license applicants. And when we look at the replacement section in it, we see that it's inadequate, confusing, and illogical at best. The section on replacement focuses primarily on the justification of the use of animals, with the first suggestion being for the applicant to justify why animals are required and perform a systematic review of animal studies. There's no actual advice for looking for replacements or avoiding animal use, and they defer to the AWERB and the named information officers to assist the applicant in looking for databases and websites. So we had a look at other sources of guidance, and they're similarly focused in the wrong direction, including our own, sadly. Focus tends to be on the other two R's rather than alternatives, despite some guidance stating it is specifically for replacement. It encourages users to explore literature of animal-based or in vivo research and recommends carrying out systematic reviews, much like the ASVU guidance. Potential replacements of animal use will not likely be found in vivo papers, although you'll find refinement and reduction. And a systematic review represents an enormous piece of work, which is a project really in its own right. And this is kind of a prohibitive use of time, energy, and resources, and is unrealistic for an individual researcher looking to see if any tech exists to replace all or any part of their research program. So there is a gap there. There's a real gap there. So replacement is poorly addressed for a number of reasons. Those listed on this slide, they're not exhaustive, but they explain quite a bit of the inertia in the system. I mean, we constantly hear that the R of replacement is the trickiest of the three. Refinement is comparatively straightforward once you have an idea of what is important for an animal's well-being. And reduction is predominantly a case of robust experimental design, but obviously I'm simplifying here. But replacement is a very different way of thinking. There's a widespread lack of knowledge of where to look for it and how to look. And this is not helped by the fact that there's no clear labeling system. So authors and databases rarely use alternatives, NATS or NAMS terminology in their tags, titles, abstracts, or keywords in papers. So unsurprisingly, there's a lack of confidence in addressing it and questioning it. It's often subject specific, but at the same time invites and requires cross-disciplinary solutions, which is not something that our brains are used to dealing with. It often means adopting new technology, which is never straightforward for us humans. And we've seen that guidance is limited and at best misdirecting. So we're often focusing on the methodology rather than thinking about the goal of the research question itself. More focus on the research question will open up different approaches to get there. So an analogy would be like it's focusing on a particular mode of transport rather than looking at the destination on a map and then working out how to get there and whether it's worth going there in the first place. So we therefore need to provide knowledge of where to look, what to look for, how to search, who to ask for help, and what is really needed to reach research goals, and how to assess existing tech and approaches for suitability. We also need to provide preparation for people to be accepting of different approaches, to be able to adopt them and adapt them to their needs. And we need them to record, report, and share their searches with others, and to be able to rethink or reframe research questions so that they select the appropriate model to answer their research question and ideally avoid animal use altogether. But overall, it's got to be accessible and easy to understand. So we're in the process of providing guidance for researchers along all of these themes, but also thinking about those who review proposed research involving animals. So whether they're members of animal welfare and ethical review bodies, AWERBs, or members of grant reviewing panels. But we need to keep in mind what the appropriate format or formats might be, what's going to be helpful to the community, what's going to be used. There's no point us putting loads of resources into developing materials that are just going to sit there. So it has to be practicable, it has to be useful for the community. So how do we know what's really needed? So we've been talking to different AWEB members, different stakeholders within the research community, researchers themselves, project license holders, project license applicants, those not using animals, those who have stopped using animals, those who've managed to transition away, also PhD students, animal techs, named persons, other training providers as well, and other 1R and 3Rs organisations, just to get a really good flavour of what is out there and what is really needed. So over the next few slides, we're going to be taking a look at the kind of content we're developing to help researchers specifically. I'm not going to go into too much detail on any of these slides, just an overview of the kind of content we're producing at the moment. And of course, knowing where to look is key. And we'll provide an updated list of active databases in alternatives, in the 3Rs, and more general biosciences as well. We'll also discuss grey literature, what that is, where to find it, and encourage users to contact biotech companies themselves. And most of the information we have on databases are taken from the Noricopa's website. A lot of those listed there are inactive or no longer supported. So we're making sure the information we provide is going to be up to date. Knowing what to look for is equally important as knowing where to look. So subject-specific terms are needed, but there are ways to maximize coverage with individual keywords, like the use of wildcards and stemming. So we'll provide a detailed guide of how to conduct searches. So we'll give examples of primary and secondary terms to combine, and how so we can help those who are completely new to this area. So for both full replacement and for partial replacement as well. There's input from AWEBs has suggested that showing examples of partial replacements for a stepwise transition away from the use of animals will be helpful. So we're going to include these too. Knowing how to search by combining these search terms using filters and operators will also help widen or narrow searches. And defining the relationship between these terms is also important to establish a robust search. So the use of filters and operators, whether they're Boolean or non-Boolean, we will go over. Different databases use different syntax as well, so that might be helpful to promote a little bit of information about the use of things like parentheses and combining those with different operators in order to create a search string. And search strings can be adapted for different databases based on the different syntax, and copied and saved and used again. And the saving of these search strings and searches is going to be a key element of the training that we're going to provide. Obviously, searching is a trial and error process, and depending on the subject area, there might be many different iterations of a search string needed to retrieve appropriate information, which is why it's one of the reasons why it's so important to keep a record of what you're looking for and where and when. So saving searches, as I said, will be a theme. Once searches have been performed with a search string, if they're saved, along with the output of the searches, they can be stored for future use and shared. Sharing is important, and we'll discuss this a little bit more later. Knowing who to ask for assistance is equally important. So it might seem quite obvious to suggest visiting the library website, but contacting librarians, they are really helpful. They're experts in searching for information, and can also signpost to different training opportunities for searching as well. Online forums and communities also exist, such as the Animal Free Research Community of Practice, and they're becoming hubs of information and ways to contact experts in the fields. Attending webinars and conferences, just like this one, reaching out to peers on social media also helps make contact with experts, And it's also worth considering getting in touch with organisations like FRAME, like Animal Free Research UK, Interniche, the NC3Rs. I mean, this is why we exist. We exist to help people move away from animal use. So it's worth dropping us a line. And knowing what you really need, I suppose, is where we begin to think a bit differently about the research question itself. So what is the ultimate goal of the research? And what is the minimum data you need to reach that goal? So to generate the data that you require, what are the minimum components you need within your experiment? And how do you access those components? Do you need absolutely all of them? Are they species appropriate? These are lots of different questions to ask. So in this particular example, we're considering the body of a mouse, which is obviously made up of lots of different organs. Which of these are important to your research? Within these organs, there are many different cell types. Which of these different cell types are vital to your experiment? With your research goal in mind, would it be more tractable to replace these cell types with human cells, or is the focus now specific in this particular example? Could those cell types be combined into a single platform or across multiple platforms using multiple techniques? Deconstructing a research question into its basic elements is really important when considering animal replacement opportunities. And knowing what you really need is not a new idea, really. This was a paper published in 2017, which suggested using a mechanistic or reductionist approach to the replacement of animals. For example, during drug development, only 3 pieces of information are absolutely needed if a drug is to progress through to the clinical trial processes of the drug development pipeline. So the first piece of information you need is whether the drug is present at the target site for the desired period of time. The second piece of information you need is the drug interacting with the target protein? And the third piece of information you need is the drug having the desired effect on the target physiological system? And to answer these 3 questions, a whole body system isn't needed. These are readily answered through molecular, cellular, and microphysiological techniques. The important thing to keep in mind is what are the minimum components needed of an experiment that would answer these questions. And knowing what you really need should also involve other stakeholders. So in many fields, it's already recognised that the cooperation of a broad range of stakeholders leads to a much more successful development, implementation and identification of applicable non-animal methods. Within clinically relevant research, so end users such as patients or clinicians are required to be consulted before a research program is developed. In toxicology, regulators are important with regards to toxicological risk assessment. They can help identify the relevant problems, after which researchers can then investigate whether a non-animal approach is possible. Experts, as well as end users, are also relevant to be included in this. Obviously, an expert in non-animal methods, for example, whether they're human relevant science or a bioinformatician, can help specify the particular research question and provide valuable input in reformulating an animal-based research question into a non-animal-based one. Information specialists are also really skilled at searching published and grey literature. Information specialists, they can help break down a scientific question into key components and base an extensive search strategy on them. Experts in artificial intelligence and mathematics can also help by providing techniques using big data or search algorithms. And we're finding now that AI is becoming a very helpful tool in identifying and labeling existing literature with NAMs and NATS terminology, so it makes it easier to find. So artificial intelligence is going to help the search become a lot more straightforward in future. It's often useful to carry out thought experiments as well. And I've used this one in previous sessions in different aspects of my career. But it's also always worthwhile asking, if using animals just simply wasn't possible anymore, how would you approach your research? What would change about your research? What would change about the ultimate goal, the question that you wanted to answer? Would you have to reformulate it into being a related question, or would you take it in a slightly different direction? What would change if you simply could not use animals anymore? So our material is going to include things like hints and tips for carrying out these thought experiments. It's also important to think about model choice as well. Now, this paper was from 2020, and the authors aimed to provide an account of criteria for organism choice derived from publications, and to allow them to develop a deeper understanding of what are the conditions associated with research using different organisms, whether they might be microbes, plants, or animals, whatever they might be, and how those choices, what influences those choices, and how that affects research outcomes. So it turns out that choosing a model organism, whether it's a microbe, a plant, or an animal, is apparently based on a set of 20 different identified criteria that include factors such as institutional support, financial considerations, and even the researcher's emotional attachment to a particular species. Some of these criteria appear to contradict each other, like the previous use of the model or novel use of the model. And so selecting a model organism involves evaluating trade-offs and weighing the importance of each of these criteria against each other, which is dependent on local context of that particular researcher or research group and their situation. It's also important to remember that all models have limitations, whether they're animals or whether they are animal-free. They're all used as a proxy of the real-life situation, which allows researchers to control certain parameters and variables and enables measurements and observations from a limited subpopulation that's then supposed to represent either an individual animal, whether human or non-human, a population or a group, or even a particular biological phenomenon. Models are a glimpse into any given situation, and extrapolation of data derived from them to reality comes with caveats, and we need to recognize these and be honest about them all. It should be the research question that drives their selection, and research needs the appropriate model to answer the appropriate research question. So we need to bear these limitations in mind when we compare the results of a search for alternatives to current model use. So when searches are carried out, does any of the retrieved information cover the minimum elements of what is actually needed? Can they replace part of the work? Is it possible to adapt or optimize protocols to make them more applicable? It's important to remember that one-to-one replacement is quite rare, but a combination of approaches might collectively answer the same question. And when thinking about achieving research goals in a different way, it's tempting to use platitudes like thinking outside of the box or turning the problem upside down. But these aren't really helpful. What we need is creative problem solving, using elements of novelty, whether they're new techniques or applying existing techniques to new situations, requires two different types of thinking, both convergent and divergent thinking. And we tend not to be particularly good at thinking about the way we think. So I'll just go over those in a little bit more detail. So with convergent thinking, it logically identifies a solution from a list of knowns. It's linear and it's systematic, and it narrows down multiple ideas into single solutions. It's focused and it leaves little room for uncertainty and comes up with what's best. So the more knowledge we have of a particular subject, the more accurately we're able to answer a clearly defined question. The convergent thinking is very similar to a systematic review, really. It makes the best decisions or takes the best options from available information. Divergent thinking, on the other hand, involves coming up with potential solutions to problems that deviate from commonly used or existing strategies. So within the scientific context, this might involve thinking about using techniques or research tools that are employed in completely different fields to your own. Thinking divergently expands conversations and trains of thought outwards. So it generates multiple ideas in the same way that you do during like a brainstorming session within your team. It then enables connections to be formed between those ideas and enables barriers to be identified, which can then become design opportunities when situations are reframed. And it can be a flexible and iterative process, thinking through the problem through different routes within a network that is created, which can then be open-ended, involving creative risk-taking. So solutions produced by this type of thinking are often quite unique and surprising. But when it comes to science, we need the best of both worlds, really. It's sometimes assumed that convergent thinking on its own should be avoided as complex problems often require novel solutions. But using divergent thinking on its own might come up with unrealistic solutions. So we need them both. We need convergent thinking to first understand the problem before applying more creative divergent thinking. So in the real world, teams will use divergent thinking to generate multiple ideas, followed by convergent thinking to analyze and narrow down those ideas, then later use divergent thinking again to come up with fresh perspectives on that shortlist, followed by convergent thinking again. And it's an ongoing iterative cycle. So thinking in a different way is very useful to identifying creative solutions to scientific problems. But we also need to be prepared to adapt existing technology. And part of the training that we'll provide at Frame will be encouraging users to become end user developers. We need to be prepared to adapt existing technology to our own requirements. And this is nothing new, really. We're all end user developers of commonly used technology, like Microsoft Excel, for example. We would all struggle to use Excel for our own requirements and purposes if we didn't first customize the spreadsheet to our own needs, whether that's formatting or function. And without that adaptation, we just can't use it. It's not fit for our purpose. So learning how to adapt is a skill that we learn and we continue to learn as the app itself develops. And this is the same with technologies. So when adapting new or existing tech to our purposes, we need to reach out to other users, to providers and developers. There might be easy to follow how-to guides available. There might be FAQs. There might be YouTube videos to help you out. So if we go back to our searches for what is already out there or what also might be applicable, when we complete searches for alternatives, we should save them, obviously. And when we've assessed their output, that needs to be saved too. And to be honest, it should all be reported. This information needs to be shared, especially if we're considering using animals. Reports of searches for replacement should be sent to an institution's AWeB for their reference, whether a researcher is applying for a project license or not. You can also report searches via email or blog about them, share them with social media, share them with peers, because others may have found something that you hadn't, and vice versa. So that was covering the guidance, the kind of guidance we're going to be producing for researchers. Now we'll move on to the kind of content we're developing to help those who are reviewing applications to use animals, whether these are applications for funding or applications for a project license. There are a number of things that we need to review and questions that need to be asked to be reassured that an applicant has fully explored the potential to replace animals in their research. So this is guidance for reviewers. This is still very much in development, but this originally was an infographic, a nudge, if you will, a reminder of what to look for when an applicant mentions possibilities of replacement in their work. So on a really basic level, looking for the what, the where, the how, and the when of those searches that an applicant might have performed could help assess the extent of their effort to search for alternatives. Did they provide any information on any of these? Were their conclusions about replacement opportunities valid? If the applicant has provided all of this information, it should make the reviewer's job a lot easier. If they haven't provided this information, then these are the kinds of questions that should be asked of them. So we're also translating these kind of questions into a checklist for reviewing research searches. Reviewers like a checklist. It helps them remember whether they've looked for everything that they are tasked to consider within an application. So we've come up with this kind of list of questions about what subject area was searched, when were the articles published and the search completed, was the search repeated again, how was it conducted, was a search string used, was that search string reported, where was the information obtained from, what databases were used, what websites, which networks or communities did the applicant reach out to, was the output thoroughly evaluated? So were the results provided within a report? Were the results relevant to the field? Was anything missing? If the results were rejected outright as being inappropriate for the use in the research, was that rejection justified? So, but it's always important to remember that if an application falls way short of being able to answer these questions, then it's worth asking what kind of support the applicant was given in the first place. If there is a gap in training, then they should then be signposted to support. So always ask yourself, was an applicant made aware of what was expected of them? Did the applicant receive any clear instruction or training about how to search and how to think about avoiding animal use? So what are the next steps for us at Frame? Well, we have a training and education working group set up to, at the moment, was tasked at looking at current training provision and coverage a little bit further to find out what is out there, what is missing, and what is worth sharing and amplifying through our own channels. We're also involved in the RSPCA's project supporting replacement in academia, which will help inform how our education and training activities develop. We will be testing these materials on AWebs and researchers and adapting it based on feedback. And we're also thinking about what's going to be the appropriate formats for nudges and for in-depth workshops, webinars, with one-to-one clinics, whatever they might be useful for the community. So on the right-hand side of this slide is a poster that we're beginning to mock up at the moment. It's the first draft of 10 top tips for finding alternatives for animals to animals in research. And this might well be the first of a series, whether they become posters themselves or postcards or infographics, we don't know yet. So we are looking for feedback and looking for a bit of guidance from you guys within a community too. We're also thinking about how we disseminate this information. And then another phase of the activity will be thinking about how we get this to funders, because obviously there's another point of review, that one of the key points of review that requires demonstration that the 3Rs have been fully implemented. So at that point, I'd like to say thanks to everyone who's helped out with this. So Adrian Smith at Noricopa, Lindsay Marshall, who's a Frame trustee and also Humane Society of the US, Renelle McLachen at the RSPCA, and the rest of the Frame team. And I think at that point, I should stop for questions. Do we have any?
Juliet, there's one question in chat just saying, are copies of your slides available?
I can send copies of the slides out as a PDF. We will also obviously be providing this recording via our YouTube channel as well. And that will be freely accessible too. Are there any other comments or questions? No, does anybody want to ask anything else? Maggie, you have your hand up. You have your hand up twice. Would you like to type your question? Or I shall try and allow your mic. Can I do that?
Yeah, can you hear me?
Yes, yes, I can.
Excellent, brilliant. Thanks, Juliet. Great talk, really thought-provoking there. You were talking earlier on about the use of AI, and it occurred to me that AI would be a much better way to select methods than getting people to do it because people are so biased.
Yeah.
Are we at the stage where we could sort of put the research question into some kind of AI system, and it would then come up and say, this is the sort of model you want to use for this work? Otherwise, I can see it's going to be very difficult to get people to drill down into all of those alternatives, especially because we all come to it with a bias that animals is our starting point. We almost need to take the human decision out of it, the human factor out of the decision. Where are we there?
It's a very good question, and I am not an expert in AI at all, but it is coming. Discussions with colleagues at ATLA have thrown up a paper that's coming out in publication soon about training machine learning tools to identify different publications within different databases to suggest them as a different way of working. But of course, AI comes with its own caveats that it maintains biases based on input data in, governs data out. So there are still going to be limitations there. But yes, it will facilitate searching, and it is already starting to.
Good.
So
good.
Yeah, I'm hopeful.
Great, thank you.
Are there any other questions?
Jenny's got a hand up. Shall I?
Jenny, did you want to ask a question? No. I'm sorry, I'm trying to enable your mic, but I don't know if it's working or not.
No, it was just to say that we heard you in the beginning of the.
Oh, okay. Thank you.
No problem.
Malcolm, on Pandora, you both have questions. I can see mics going on and off, so I speak.
Sorry, that might have been me and Leon.
Oh. Pandora, Mike, Malcolm.
Yeah, really good talk. Thanks, Juliet. Can you hear me?
Thank you. Yes, I can.
I was just thinking it's the point at which this guidance is given is really important, because if it waits until it's right down to the level of the project license application, then by that stage, the project's already been funded, it's gone through the AWERB, and it seems to me it's going to be really hard to turn that around at that stage. And And so it would just appear that targeting funders and making sure that people think about these issues right at the point at which they're, formulating their question and gaining funding seems to be the place to start, because then, then it's not sort of each process you go through, it's sort of set in stone a bit more, isn't it? Once you go through the AWERBs and you've got funding and so on. So that's just what I was thinking as you were speaking.
Yeah, I agree. I think it is important to engage with funders and talk to them about their review processes, because at the moment it does tend to be subject specific on its own, although they do have 3R's expectations within their guidance. Yeah, that it's important to shore up that process. I totally agree with you. So that is informing part of our work at the moment, and we're just trying to work out the best way of moving forward with that. I agree. Thank you. Malcolm, did you have any questions to ask or any comments to make? If we can't enable your mic, then please do type a question out. No? Amy, are you able to unmute Malcolm?
It should. His mic's been enabled. Let me just.
Sorry, it's a long question. Okay.
No. It should be enabled. It should unmute.
Yeah, Malcolm, it looks like your mic has been unmuted for me as well, so might be a quirk of the system there. So while Malcolm's typing out his enormous question, has anybody else got anything to either comment on or to ask? Or if anybody has any advice for us about how we go about disseminating this, that would be really helpful as well, please.
I really liked your idea of the thought experiment.
Ah, thank you.
I think it's so important to get people to change the question rather than the methodology.
It's thinking about what is vital to that research question, and if we're going to think about it in more extreme terms, what would we lose if you couldn't answer that research question in its current form? Malcolm, I'm sorry that we couldn't answer your question. We've now got to time. I'm afraid it's 10:45 now. So if you want to talk offline, then I'm more than happy to. I'm happy to stay on. But I think we should probably draw things to a close there. Thank you so much, everybody, for joining us this morning. When the recording is up online, we will let you know, and please feel free to share it with your own networks too. Thank you very much. Hope you all have a good day.